Last time we were talking about control of microbial growth. The time before that we were talking about the dynamics of prokaryotic growth. There's a little overlap between those two chapters if you think about it. Chapter 4, we identified the environmental factors that influence microbial growth and the nutritional factors that influence microbial growth. Well, if you think about it, if you affect any of those, the environmental factors or the nutritional factors, you can control the growth of the microbes. Refrigerating something, cooling down the microbes, controls their microbial growth. We talked about that as a means of controlling their growth. So realize that there's a little bit of overlap between those two chapters with regards to the material. Any questions with regards to chapter 5? Okay, so tonight we're going to move into one of my more favorite chapters. We're going to talk about metabolism. This is chapter 6. When we talk about metabolism, we're really talking about the cell growing. And in order for the cell to grow, it's got to do two things. It's got to synthesize new products, and it's got to harvest energy. Synthesizing new products is called anabolism, and harvesting energy is called catabolism. We're really going to focus on the harvesting of energy. We're not really going to talk much about the synthesizing of molecules for the most part. When we talk about harvesting energy, really we're talking about the cells converting energy from one form into another form. So with plants, when we talk about photosynthesis, the microbes and the plants that do that, that photosynthesize, convert light energy into chemical energy. Glucose, that's chemical energy that they're producing. And then those microbes and animals that eat the glucose and metabolize the glucose are then taking that chemical energy and turning it into ATP. Now, of course, ATP is a chemical too, so it's still chemical energy, but now it's in a form that the cell can readily access. In fact, it's the only way that we can access energy is through ATP. So we take all of this chemical energy that we take in and we convert it into currency, if you will, into ATP that then can be used to do work, to synthesize new molecules, to grow and reproduce. We can talk about molecules, compound stuff having free energy, the energy that is available to do work. This is the energy that is released when bonds are broken. So anytime you break a bond in a chemical, energy is going to be released. Breaking the bonds in a glucose molecule releases energy that we are going to capture and use to do work. Now, a glucose molecule has a certain amount of energy, and we can release all that energy at once just by burning the glucose molecule. Combusting it would release all the energy that the molecule has. But that would be very useful to the cell, because it might not need all of that energy at once, and if it was all released at once, a lot of it would then be wasted. So the cell has the ability to release that energy in a stepwise fashion, if you will. Release a little bit now, capture the rest of it in a chemical bond, release a little bit later, capture the rest of it in a chemical bond. So they're able to take advantage of the energy that would be in the molecule, releasing it a little bit at a time as they need it. They do this with the use of chemical, or with enzymes. These enzymes are able to break molecules, form molecules, break molecules, form molecules, and allows them then to take advantage of the energy in the molecule. So before we get into the pathways of metabolism, I'd like to take just a moment and talk about some of the terms and some of the components of metabolism. First component we want to talk about, enzymes. Now, you've probably been exposed to enzymes somewhat already in your schooling, but I'm going to close this door, a little loud out here. So enzymes are proteins. Proteins then are coded for by the DNA. So these are molecules, molecules that the DNA codes for specifically. If you don't have the gene that codes for the enzyme, you don't have the enzyme, you don't have what the enzyme can do. Enzymes serve as catalysts. They speed up chemical reactions. They facilitate those reactions, if you will, converting substrate to product. Now, sometimes this conversion of substrate to product happens in breaking up the substrate so that you end up with two pieces, if you will, catabolism. And sometimes it's the building of products, anabolism, where you take two pieces and bring them together. In both cases, what the enzyme does is it lowers the activation energy. What this means is it lowers the amount of energy that you have to put into the reaction in order to get the reaction to occur. If you imagine a graph here, and let's say that there's so much energy here at the start of the reaction, okay? This is where the substrate might be, the thing that we're going to break up, that we're going to break the bond and release the energy to. Well, here's the thing. To break that bond, there's going to require an energy input. That energy input, without the help of an enzyme, might be substantial. But at some point, you could put in enough energy to break that bond. This is the energy we're putting in. And in doing so, release the energy in the bond, and this would be the product at the end. And you would have gathered or gained this much energy here. But it might take a while, additional energy, if you will, to cause this reaction to occur. Might be slow. With an enzyme, they lower the activation energy so it happens sooner. We don't have to put in as much energy, and we still get the same end product. So we still get the same amount of energy out of it, but we don't have to put in as much to initiate it. The enzyme lowers the activation energy in order for the reaction to occur. A couple of other things that we can say about enzymes. They are specific. An enzyme only works with one molecule. So if an enzyme works to break up a glucose molecule at a specific spot, that's all that enzyme does, is break that glucose molecule at that specific spot. So you need an enzyme for each and every chemical reaction that's going to take place. The other thing is that enzymes are conserved. We don't have to make a new enzyme for each and every reaction. An enzyme can cause a reaction to occur and then move on to new substrate and cause the reaction to occur there, and move on to the new substrate. And so the enzyme will cause the reaction to occur until all the substrate has been used up. Any questions about enzymes? Let's talk about ATP. This is the energy currency that we're going to produce. The whole reason for breaking down our molecules. This is what we can spend in order to do work. When we look at the ATP molecule, it's an adenosine triphosphate. It's an adenosine molecule with 3 phosphate groups on it. But the bonds between these phosphate groups are high energy bonds, represented by a little swiggle here. When work is being done, we can break this bond here, releasing a phosphate and releasing energy, leaving adenosine diphosphate which we will use when we are breaking glucose down to reestablish ATP. So when we put energy in, and we'll see how we do this. We can do it through a process called we'll do it through a process called substrate phosphorylation or substrate level phosphorylation. In substrate level phosphorylation, what's happening here is we are generating an ATP molecule directly from the breaking of the substrate. So when we break a glucose molecule, for instance, if we were able to generate ATP directly from the breaking of that molecule, that is substrate level phosphorylation. Anytime you break a molecule and that energy from that bond being broken is captured in ATP directly, that's substrate level phosphorylation. I tell you that because there is also a process called oxidative phosphorylation. And oxidative phosphorylation involves the proton motive force. We haven't talked about that yet. We will just here shortly. But the proton motive force establishes a hydrogen ion gradient that generates the electricity. So as the energy. So as the hydrogen ions move with the gradient, they'll generate ATP. So we'll see how that happens with the proton motive force. Any questions about ATP?
Can we go over the substrate level phosphorylation again?
Okay, so anytime we have a molecule, so suppose we have glucose, and we're going to break this glucose into two, it's a 6-carbon molecule, right? And we're going to break it into 2 3-carbon molecules. In doing that, if we generate ATP directly from the breaking of this molecule, that is going to be substrate level phosphorylation. Because the ATP was generated from the energy released when we broke the bond of the molecule itself. Okay, so ATP is coming directly from the breaking of that bond. So when we talk about our energy source, we're referring to our fuel. We're talking about the compound that's going to be broken down. Okay? For most of us, we're talking about, for most of our cells, we're going to be talking about glucose. We'll see that bacteria also break down a lot of other compounds, but anything that the cell breaks down that it uses as fuel serves as its energy source. This requires then an oxidation-reduction reaction, a redox reaction. In a redox reaction, what we're doing is we're moving an electron from one molecule to another molecule. So here's molecule A in the reaction. This is molecule B in the reaction. And molecule A has the high energy electron, or the electron that's going to be moved. So in this reaction, A loses the electron. It's been oxidized. And B gains the electron. It's been reduced. Which molecule has more energy at the end of this reaction?
B.
B does, exactly. This electron here is a high energy electron, we'll say, and A, in being oxidized, loses that energy. So its energy state will be lower, and B, in becoming reduced, has gained that electron, and so its energy state will be higher. What's the charge of an electron? It's negative, isn't it? What are the ways, whenever you add a negative to something, what are you doing? You're reducing it. You're reducing it. One of the ways to remember this concept here is that the electron,